Hi my dear students, welcome back to Tungal online classes. In the last class we had discussed regarding principles of biotechnology. Two main techniques we had discussed in the last class. The first one is genetic engineering and second one is maintain the sterile medium in ambience beside the surroundings. Here for example, first one, what is genetic engineering? Genetic engineering which alter the chemistry or chemical nature of nucleic acid is nothing but DNA and RNA. And second technique also very important. Why? Because if you are not maintaining sterile or non-microbial region, then what happen? Unwanted bacteria as well as some of the fungi also be going to grow on that medium. The medium is going to contaminated. Further, we will not get the desired character, desired gene product we are not going to get. So that's why we have to maintain the sterile medium in surroundings. Okay, and also we discuss regarding traditional hybridization. The farmers use a traditional hybridization to promote the desired characters from one generation to another generation. Then here one demerit is here. What is the demerit? Beside the wanted characters, unwanted characters also going to transfer to the next generation. That problem is solved by using of modern biotechnology. In the last class we had discussed how it is going to solve the problem. And in last class we discussed one question like, what is the fate of unknown or desired DNA in host cell of cytoplasm? It is not going to, if simply we are insert the desired DNA in host cell of cytoplasm, it is not going to get replicated. Why it is not going to get replicated? In the last class we discussed now. Why? Because the desired DNA, it is not a part of genome of host cell, so that's why it is not replicated. When it is replicated, the desired DNA we have to paste, it should be a part of genome of host cell, then only it is going to replicated. Clear? And one more reason is given. During the replication of chromosome or DNA, replication is start with one special sequence that is called as origin of replication, ORI sites. Such ORI sites are absent in where? Desired gene. So that's why it is not get doubled or not active in cytoplasm. Clear? And this all information we discussed in the yesterday, and we prepared like artificial chromosome or artificial DNA, like how we are going to prepare, we discussed. And today I am going to start with the tools of recombinant DNA technology, very important topic. Here in biotechnology, now we are going to discuss regarding tools of a recombination technology, tools of rDNA technology or recombination DNA technology. Which tools we needed? Here the main key tools are the first, we need enzymes. We need enzymes for cutting as well as pasting, first thing. And second, we need vectors. Vector is nothing but plasmid, which is useful to transfer the genetic information from one cell to another cell. And we need one donor cell for isolation of desired like insulin genes. And one more cell we need, that is host cell, for replication or for production of clone cells. And the last one is bioreactors, which is useful for purification of gene product. So this is a main key tools for done the rDNA technology. Let's write the note. The first which are required, enzymes. Without enzymes we can't prepare rDNA, you know very well. And here enzymes are two enzymes in must and should, the first and second. First enzyme is restriction enzymes, restriction endonuclease enzymes. restriction enzymes we needed. Purpose is what? The purpose is for cut the DNA in one specific location. And second, one more enzyme is ligase or ligases. It is required. Why it is needed? To sticky, to attach the two different fragments, to sticky the DNA or to attach to paste the DNAs, to joining of two DNA fragments. First tool. And second one is vector. Here vector is nothing but what? Plasmids. Vector here nothing but what? Plasmids. These plasmids are used for transfer of genetic information from one cell to another cell. Transfer of genetic information from one cell to another cell. This is second one, compulsory we need. Okay, and third one is one donor cell. For what? For isolation of DNA. For isolation of desired DNA, nothing but wanted DNA, which character you want to develop. Clear? Next, and one more cell is required, that is host cell. In host cell, this desired DNA is going to get a double. The doubling is nothing but what? Clones. This clone word we already used in second year in the first chapter. Asexual reproduction. During the asexual reproduction, the formation of progenies are exactly equal to the mother. So such identical offsprings we call as what? Clones. Here also clones are formed. Which clones? Desired DNA clones. Just Xerox copies are going to form. Get doubled in host cell. Mostly here we are going to use prokaryotic cells. Mostly prokaryotic cells we are using, and sometime eukaryotic cells also sometime we are going to use. Clear? Next, now these desired genes are expressed in a host cell. Now the desired gene product is formed in host cell. Now we have to obtain, for that, bioreactors. Bioreactors are needed. Bioreactors are which are used for what? Purification of gene product. Purification of that desired gene product. Okay, so this is actually main tools for preparation of DNA technology or preparation of recombinant DNA. Got it? Sure. The first we are going to discuss regarding enzymes. According to the NCERT, we are going to discuss the first one is restriction enzymes. Just put the heading. The first one is, the major tool is restriction enzymes. Restriction enzymes. In NCERT, they given generally in the 1963, the scientists have recognized two enzymes in one bacteria, that is nothing but E. coli bacteria. So these two enzymes are responsible for restriction growth of bacteriophage within the bacteria. Okay, so these two enzymes are or nothing but the two enzymes are belong to restriction enzymes. The first component of restriction enzyme is methylation enzyme, and second enzyme is nuclease, nothing but restriction enzymes. Okay, see, first write it in a the scientists are in the 1963 the scientists are recognized two enzymes are noticed in E. coli bacteria, So these two enzymes are responsible for restriction growth of bacteriophage, nothing but RNA in bacteria. It means these two enzymes, they are defense mechanism of bacteria. So due to presence of these two enzymes, the bacteria is going to save from attacking of virus or bacteriophages. Clear? So that's why these two enzymes also we can call as antibiotic resistance compounds. These two enzymes also act as immune system of bacteria. So this immune system, it is going to save the bacteria from virus attack. So here the virus, it is not going to replicate in this E. coli bacteria because it is consist of two components or two enzymes. They are, the first enzyme is for methylation of bacteria genome, and second enzyme for fragmentation of virus RNA or DNA, and the second enzyme for fragmentation of virus DNA or RNA. Here one point, the question is here, virus do not consist both nucleic acid. No virus tell it containing two nucleic acid. It should be containing either DNA or RNA. If it is a plant virus, it is containing RNA, and if it is animal virus, it means it causes the disease to animal, that is called as animal viruses. Genetic information is DNA, but two nucleic acids are never present in virus. Got it? Clear? Sure. Next, now this second one is useful for fragmentation of virus DNA or RNA. So that's why virus RNA is not replicated in host cell like E. coli. Clear? Which enzyme that one is? The enzyme is restriction enzymes. The enzyme is restriction enzyme, which have the capacity to recognize unknown particle or unknown RNA. If it is entered into the host, it is going to fragment the unknown DNA or RNA. Here unknown DNA, RNA is nothing but what? Virus containing DNA or virus containing RNA. Due to that, it going to kill. Clear? So this is actually the scientists have noticed. Got the point or not? Next, here the question is comes, sir, why it is going to attack only on unknown DNA or virus DNA? The restriction enzyme means what? It is going to cut the DNA. Then why it is not going to cut the own genome of bacteria? That is actually question comes in mind. The question is what? Sir, why a restriction enzyme, why the restriction enzyme is not cutted or not fragment the its own genome, cut the own genome. Clear? Simple. See here, this is one bacteria cell. Here bacteria genome, nothing but this is a chromosome, single chromosome present. Based of this, two components are present. This is one component and this is one component. So this one component nothing but enzyme which is useful for methylation, and this is second one is enzyme is nothing but a restriction, nothing but restriction. Now what happened? For example, one virus is going to attack. One virus is attack the bacteria. Now this is nothing but bacteria phases. One of the bacteria phase, nothing but virus is entered. And here this is a virus. Virus DNA is entered. Now here this is the bacterial genome. Here one component, here one component. This component is what? A restriction enzyme. Now it is going to cut this virus DNA or RNA. Now this enzyme is going to react on. Now it is going to become fragment. It is going to become as what? Fragmentated. It is going to kill. Our question is what? Sir, why this is not act on bacteria containing genome? Bacteria containing genome. So this is actually our question. The answer is here what? Here the answer is, we said now, first one is what? For one methylation purpose, one component or one enzyme is there. Got it? Here this component is methylation, methylated the genome of its own bacteria. Then again question, sir, what is methylation? Methylation means nothing but adding of methane CH3 groups to restriction site where the restriction enzyme is going to cut the DNA. Got it? Adding of methyl group to a restriction site. Chalo. Now what happened? When we are adding of methyl group to restriction site, the chemical nature of restriction site is changed. That changing restriction site is not recognized by restriction endonuclease enzymes. So they are not going to identify. The restriction site. If they are not going to identify the restriction site, it is not going to cut the DNA. It is not cut the own genome. Got my point? How bacteria genome is saved from restriction enzyme? The answer is only one, sir, because it is already methylated. Methylated means adding of methyl groups to it where restriction site. So due to that, the restriction enzyme is not going to recognize the restriction site, so it is not going to cut the DNA. But in this case what happened? So this viral RNA is not methylated, restriction site is open. Now this restriction enzyme is going to cut the virus RNA or DNA. Clear? So this is regarding restriction enzymes. Restriction and the nuclease enzymes are first discovered by Nathan Smith Arbor. So these three scientists are collectively got the Nobel Prize also. Okay, and next, and now we have nearly more than 900 restriction enzymes from nearly they are isolated from 230 bacteria or different strains of the bacteria. Okay, let's write it. This restriction enzymes are discovered by Smith, Arbor, as well as Smith, Arbor and Nathan. So these three scientists have got the Nobel Prize also. They shared the Nobel Prize for discovery and study of restriction enzymes. First, and second point. Now we have nearly more than 900 restriction enzymes. These isolated from nearly 230 different strains of bacteria. Different strain of bacteria or different type of bacteria. Okay, and this each and every enzyme have the capacity to recognize a specific and particular or unique sequence. Okay, next, these enzymes can recognize specific sequence of DNA. So 900 enzyme means 900 specific restriction enzymes, the restriction site they are going to recognize. Okay, and next one, restriction enzyme is Hind2, Roman number 2. So this is the first restriction enzyme. Okay, so based on this, a number of enzymes are also be there, like ECO R1, like enzymes. Actually here what is HIND and what is ECO R1? Actually it is a nomenclature of enzyme. It is a nomenclature of enzyme. Let's write it. In nomenclature of enzyme, here the first letter is denote genus of the species where we isolated this enzyme. Second one is species, and third one is type of the cell, and last one is the Roman number is indicated a serial number of enzyme, and here it is which type of enzyme it is going to tell. Okay, let's write nomenclature of restriction enzymes. For example, ECO R1. ECO R1 means what? First letter E. E means what? It is the meaning of E is, it is a genus of bacteria. Genus of bacteria. Next letter is CO. E means what? Escherichia. Escherichia. Actually, this is the meaning. And next one is what? CO. CO means species. Species, name of bacteria from which location we isolated. Next one is R. R means it is a type of strain or nothing but type of bacteria. It is to indicate the character of bacteria. And last Roman numbers are here. It is a serial number of restriction enzyme, and also it is indicated a type of enzymes. Clear? Mainly three types of enzymes are there: type 1, type 2, type 3. Restriction enzymes type 1, type 2, type 3, Roman number. Mainly in recombination DNA technology, only type 2 enzymes are used. The question is why? Because if you use the second type enzyme, there is no need to add the additional energy to the enzyme. Okay? But in the case of first type and third type, to activate the enzymes, we have to supply the additional amount of energy. Energy is nothing but ATP, adenosine triphosphate. Okay? Let's write it. Three types. One is type 1 enzymes, type 2 enzymes, type 3 restriction type 1, 2 and 3. Here mainly restriction type 2 are used because there is no need to give or to produce, to supply ATPs. Okay, next. Here a restriction endonuclease enzymes. Nuclease, it is a broad class of enzymes. These enzymes are belong to one broad category, that is nuclease. Nuclease, ase means what? Active. The enzyme, it is going to cut the information which is present within the nucleus. Okay, here belong to nuclease. They are belong to which class? Nuclease. Nuclease enzymes are mainly three types. The first enzyme is exonuclease. Second one is endonuclease. Third one is restriction endonuclease enzyme. Okay, let's see. First one is what? Exonuclease enzyme. Here it is going to cut the DNA or remove the nucleotides at the end of the end or terminal site of the DNA. Second one is endonuclease enzyme, so which have the capacity to cut the DNA at the middle of the DNA. So these two enzymes are cut the only single strand of the DNA. And third one is restriction endonuclease enzymes. It has the capacity to cut both sides of the DNA in one particular sequence or particular recognition site. Let's try it. First one is exonuclease enzyme, which is going to cut the DNA or remove the nucleotides at a terminal site of DNA. Terminal site of DNA. It is going to remove the nucleotides at the terminal site of the DNA. Only one single strand is going to cut. Next one is endonuclease enzyme. Here it is going to remove the nucleotides at the center or middle of the DNA. center of DNA. Okay, let's see diagram. So this is one DNA sequence. If you use exonuclease enzyme, it going to remove. So this here one nucleotide is here na, that is going to cut. Remaining is like this. So this is first exonuclease, and second one is endonuclease. It going to cut the DNA at a middle of the DNA. Here it going to cut. We can remove the nucleotides from middle of the DNA. Second, and third one is a restriction endonuclease enzyme. Special enzyme which are mainly used in DNA technology. See here in this enzyme, the first it going to inspect the entire sequence or entire length of the DNA first, and second it going to recognize the restriction site. Once it is recognized restriction site, it going to bind on that site. After binding, it going to cut the DNA at both sides in a particular fashion. Okay, let's write. It going to read complete length of DNA first. Second, it is recognize some special or particular sequence or unique sequence that is called as recognition site. They recognize recognition site. Recognition site is nothing but a restriction site. Then let what happen? It going to bind on this recognition site. Recognition site. Next, it is going to cut in a particular fashion. It is going to cut the both DNA stands at a particular fashion. Okay, so here the point is, sir, where it is going to cut. The DNA is a combination of long polynucleotide double stranded. Where it is exactly going to cut? The question is here, which is the backbone of the DNA? Which bonds are backbone of the DNA? Which are phosphodiester bonds are backbone of the DNA. Now this enzyme is going to cut the backbone of DNA. The bonds are phosphodiester bonds. It is going to cut the DNA at a phosphodiester bonds. Okay. Next, sir, here you said what you said, recognition site, special or unique sequence. The special or unique sequence we can call as, generally in science language we can call as palindromic sequence. The special recognition site is called as palindromic sequence. Palindromic nucleotide sequences. Here, this palindromic nucleotide sequence are recognized by special type of restriction endonuclease enzymes. Each nearly 900 REN we already isolated. Each enzyme is going to recognize one special palindromic sequence. Actually, what is palindromic sequence? Palindromic sequence means it is a group of letters which give the same meaning while we reading from both directions. It means from forward and backward. In both direction also it gives the same meaning. Okay, such special letters we can call as palindromic. Let's write palindromic nucleotide sequence. First, write regarding palindromic definition. Palindromic means it is a group of letters which give same meaning, or same word from while reading from forward and as well as backward. If read like this or if we read like this also, it gives same meaning as well as same word. That is called as what? Palindromic words. Let's say example. Palindromic examples are, for example, madam, M-A-D-A-M, madam. Just read it from here to here, from this side, M-A-D-A-M, madam. If you read in opposite direction from backward, again M-A-D-A-M, madam. Same meaning is coming. For example, wow or level. From this direction or this direction, same meaning, same word. Level also. These are called as what? Palindromic words. In biology, how we are going to? Here, palindromic in DNA. It is a sequence of nucleotide on both strands which give the same meaning while reading backward to forward or forward to backward. That is actually palindromic in biology. Write it. Palindromic in DNA. It is a sequence of nucleotide on both strands of DNA which give same sequence, nothing but same meaning or same word from both direction, one forward and one backward. So for example, take one restriction endonuclease enzyme. Which one? ECOR1. This is one restriction endonuclease enzyme. So this have the capacity to recognize one special sequence, nothing but palindromic sequence. So let's take one long stretch of DNA. This is a long stretch of DNA. And you know that here adenine is always going to bind with thymine. Guanine is always going to bind with cytosine. Nucleotide base pairs we already discussed in 11th class. Okay, let's see. Here for example A, T, C, C, A, or here thymine, here adenine, here guanine, and here thymine. Here like, so this is one long stretch of DNA. Now we are using which enzyme? EcoR1 enzyme is used. Now this restriction endonuclease enzyme, first step, it going to read the entire sequence of DNA, first point. Second, it recognize restriction site, nothing but palindromic site. What is palindromic site here? Which give the same meaning. For example, here, take this portion, special portion. If you read from this direction, in this direction, GAATTC, it is actually a nucleotide sequence. Again you read from opposite, GAATTC. So this sequence is a special sequence, we can call as what? Palindromic sequence. So this sequence is recognized by which? EcoR1 enzyme. Other restriction enzyme have no capacity to recognize this sequence. Got the point or not? Next what? It going to here attach. This is the enzyme. Here this will also be enzyme. It going to attach on that sides and cut little away from the palindromic sequence. Little away here. From here palindromic sequence, but it is cut here in between G to A. On both strands of same nucleotides or same sequence of nucleotide, again it going to cut in between G to A in a one particular fashion, like it going to cut. Okay. Now due to that what happened? Due to that the DNA is going to cut it to form two overhanging strands are formed. So these overhanging strands are containing free hydrogen bonds. Okay, see here how it is going to cut. Here like it is going to cut. It means here which is going to cut? Here which is there? Hydrogen bonds are there. So these hydrogen bonds are going to cleave. Nothing but the cutting is occurred at a phosphodiester bonds and also between two nitrogen bases of hydrogen bonds. Okay. Now this is actually activity of restriction endonuclease enzyme. Okay, let's see how it is going to form. Take only one, this one is GAATTC and same in GAATTC. It is cutted here, it going to cut on both stand of same nucleotides to form here one is G. This is one small stretch and here C T T A G plus one more strand is what? A T T C and here only G. Like it is going to form, like two strands are formed. Here each one overhanging is consist of hydrogen bonds which is useful for paste the or sticky the desired gene by using of ligase enzyme. Okay. So this is actually how the restriction endonuclease enzyme is going to work and cleaves the restriction site, and to form this strand is called as what? This strand is called as sticking strands. Overhanging strands are formed, free hydrogen strands are formed, that is called as what? Sticky strands. Okay, let's say for example here take one plasmid, here plasmid. Here take here human, here one human genome. This is one long stretch of DNA, long stretch of DNA. Then you have to use same enzyme for cutting off plasmid as well as cutting off human genome, nothing but human DNA. Same enzymes are used. Here EcoR1 is here used. Here same EcoR1 enzyme I used. Okay, then what happened? It's going to recognize one specific sequence, polynomic sequence. I'm going to cut like sun is going to form. And here also same enzyme is used now, but this is very long stretch here. Here one restriction sequence is there, polynomic. Here also one sequence is there. Here also one sequence is there. It means cleavage 1, cleavage 2, cleavage 3. And here 3 times it is going to cleave. To form how many pieces? 4 pieces. Like like 1, 2, 3 and 4 pieces are formed. Okay, now simple, what you have to do? So these which fragment you want, that fragment we have to paste to the plasmid. Now this fragment we are going to paste to vector, nothing but plasmid. So this is, we added a desired gene, we added. Now it is become is what? R plasmid. Now this R plasmid we are going to transfer into host cell. Now see, this desired gene, now it is a part of plasmid. When plasmid is going to get double, at the same time desired genes are also going to get double. Okay, now we can get the desired gene product in host cell. So this is actually how restriction endonuclease enzyme is used for construction of recombinant plasmid. Okay, in the next class I am going to start with how to separate this 4 fragment from the cell. We have one technique is there in the biotechnology, that is gel electrophoresis. Okay, and my dear student, the biotechnology topic is one of the very tough topic and important topic in the 12th. So you have to focus first on YouTube class first, watch it properly, and next read the NCERT. Then only you can get the understand. Okay, thank you all of you.